These are some examples of oncogenes that are known to cause cell division, to be a proto-oncogenes. You probably heard in other classes about the SARC genes and RAS genes, JUN, FOS, BCL1, MYC genes. You don't have to know those more specifically. They're just like 2 genes that are important for concept. Now, so these are some of the tumor suppressor genes that are important. Remember, tumor suppressors need 2 mutations in order to get the cancer. So if you have p53 gene that is in the nucleus, this is important for transcription factor and regulation of apoptosis. And if you have this retinoblastoma gene, also in the nucleus, is important for transcription activation, the transcription factor. So look, 2 of these here, they are very important and very commonly mutated, commonly found as mutated in cancers. That is the p53 and the retinoblastoma gene. I'll show you how they work. Now, when you have cell division, we know the cell division has to be controlled. You have control cell division. So what controls the cell division are these cyclic-dependent kinases. Cyclin-dependent kinases. And those are abbreviated CDKs, or cyclins. Now, where are they? They are in specific checkpoints. With the cell division, you have G1 to S transition. So remember I mentioned that between G1 and S, you have a checkpoint. What takes care of the checkpoint is the RB gene, is the retinoblastoma protein. And then from G2 to mitosis, you have another checkpoint, and that one, like mitosis promoting factor, I will not focus on that one that much. This one is the most important one. And then you have another checkpoint that is spindle assembly. If all your chromosomes are not lined up, then the cell does not divide. And these are the most important checkpoints for the cell division. So how is it that this transcription factor is working? This retinoblastoma gene. This here is the retinoblastoma protein. Now, this is important for every cell. So it's a bit misleading that this cell is called retinoblastoma, because when you hear it, you think about retina, right? But it was found in the retina and it was called retinoblastoma, and then the name is stuck. So that's why this is called the RB protein, but it's present in every cell. So what that protein does, it is binding a factor, a transcription factor, and is keeping it inactive. So if you have an active protein, you are inhibiting something that is going to allow the cell to divide. Okay, so that is the tumor suppressor gene. Now, when you have those cyclins working and you start to go, you're going to go through the cell division, usually you have a lot of phosphorylation events that go on. You probably saw this also in the biochemistry classes or basic biology classes. Phosphorylation, this phosphorylation controlling activity of proteins. So here you have a couple of phosphorylation events. When the retinoblastoma protein gets phosphorylated, it will dissociate from this transcription factor, and then this transcription factor will go and bind to the promoter, and then you get certain genes that are activated. Now, if you think about it, and you have a mutated retinoblastoma that does not work, you don't need to dissociate it, because if it does not work in the first place, it's never associated in the first place. So when you have one mutation here, and the 2 mutations in the 2 alleles, and all of your RB proteins are not functioning, that means this transcription here is happening all the time, because E2F factor here is free to act, is not held back by anything. Retinoblastoma protein is what is holding back the transcription factor. Now, what causes the protein to undergo cell division, or to start to the G1 stage? You have several stimuli that can cause cell division. Some of them can come from the outside of the cell, so it will bind to the membrane, some type of receptor, and then you have some signal transduction that is going to signal for cell division. Yes, you have a question? So usually with these events for signal transductions, you have phosphorylation going on. Then you know that if you have phosphorylation, you have a lot of usage of energy because you have to have those phosphate groups coming from some type of energy, ATP here in this case. And then you might be asking, why is it that you have so many steps? Why is it that you just don't get activation here and straight into the nucleus and activate cell division? Well, it's because at each of these steps you get amplification of the signal. So you have a more efficient type of message from the surface to the nucleus. You have a signal amplification effect. So the first stage in which you have a checkpoint that is very important for the cell not to be divided or to be controlled cell division is between G1 and S. Okay, so this is the G1 and S checkpoint. And here, I just told you why so many steps. Okay. I don't need to concentrate. I just told you overall what this is. To activate cyclins. Okay.
Do we know how cancer stops or inhibition stops these checkpoints from occurring?
Yes. Like in the case of retinoblastoma, if you have to have a protein that is active in order to suppress cell division, and the protein is just not active anymore, then you cannot suppress anything.
So wouldn't it be just as easy to just either supplement the protein artificially or to just, isn't there a way to just jumpstart that checkpoint again? Like it was working and it mutated, isn't there a way to just get it back into the new state?
Theoretically, yes. You can use gene therapy.
So
But
we just use that.
because how are you going to get the genes inside the cells? Usually you have to use some type of retrovirus in order to do that. And it's a matter of fixing one side and damaging another. Because some retroviruses that were used for cancer, for gene therapy before, they're working in some particular cancers, but then they cause some other cancers. So once you have a problem, it's very difficult to circumvent that problem. So as I mentioned, you have stimulatory pathways or inhibitory pathways. That the inhibitory pathways will hold off cell division. Stimulatory pathways will activate the cell division. You don't really have to know details about this here. Okay, this is an important one here. This is where the p53 gene comes along. So if you have these cyclic-dependent kinases, they are inhibitory proteins, and they need to be activated in order to stop cell division. If you have a mutation on the inhibitory protein, so you're not stopping cell division, and you just progress into cell division. Now you have the gene that is the p53 that enhances the expression of inhibitory proteins. Okay, so p53 enhances expression of inhibitory protein. So if you damage p53, that means you lose your inhibition. So now the cells are just going to divide freely. There is nothing at that specific checkpoint from G1 to S stage. There's nothing at that specific checkpoint that's going to stop the cell division. Yes, p53 will cause the expression of inhibitory proteins. Okay, so the p53, what it's doing is putting on the brakes. Okay, is enhancing the expression of these proteins that are holding off cell division.
So you don't have p53, you don't have the inhibitory.
Right, you don't have a brake pedal in there. So the cell will lose a checkpoint for stopping cell division. Okay, now let me jump here. This is just as I had mentioned before, the retinoblastoma that is inhibiting the If F2, the elongation factor for transcription. And if your retinoblastoma does not work, that means you have a constitutive expression of genes that are supposed to be regulated. Now, examples that I mentioned here for tumor suppressor genes are the P53 and retinoblastoma. Okay, those are the two that I would say, if you remember anything out of cancer genetics, you remember P53 and RB proteins. They are suppressing the progression from synthesis stage of DNA to the progress of cell division. Now, I put a little movie here that I think is important to just, relax, chill out. I'm sorry I'm driving you to 2.5 hours of intense last day thing. But, if you want to go to the restroom, get a little water, But I just want to get as much as I possibly can.
The virus is derived from a typical cold virus called an adenovirus. It infects or begins to infect all cells. If the virus infects a normal cell...
Now, this is the idea of how can you use this system of inhibition of cell cycle in order to trick the cell to commit suicide. Okay, so this is one of the ideas that you can use a virus in order to get these cancer cells very sick.
The DNA from the virus will get out of it and go to the nucleus. But then the cell has an antidote, P53 in fact, the tumor suppressor gene, which then prevents that viral DNA from replicating, and the cell lives. On the other hand...
So that happens in a normal cell. P53 is a gene that is actually involved in many aspects of the cell cycle. Is like one of those fundamental transcriptionally activating genes. So now when you have a cancer cell that has a modification on the P53, and you're trying to use this type of strategy in order to kill this cancer cell...
If the same virus infects a cancer cell, the DNA will get out from the virus, travel to the nucleus, and there's no P53 around because the P53 is mutant. It's defective. Therefore, the viral DNA will continue to multiply, will eventually burst the cell, release lots of viruses into the environment. They will infect both normal and cancer cells, but the normal cells are kind of immune to the virus, whereas the cancer cells will continue to be killed by it because they have no P53.
Cause another cancer.
That's the thing, because you are assuming that all of your normal cells have an intact working P53. And a lot of times, we're using this type of viruses, like adenovirus or something else, the virus will find a way to circumvent the cell defense. So it's, especially with the virus that goes into the nucleus, the virus that sends DNA into the nucleus, is a risky business. Okay? Now, this is actually the area that I work with, which is oncolytic viruses. But the virus that we are, like putting our money into is a virus that just stays on the cytoplasm. It does not cause mutation in the DNA of the host, and it can kill the cell in the cytoplasm. Now, if it goes into a cell that is healthy, the cell has antiviral defense mechanisms that may also involve P53, but it mainly involves interferon pathways. Okay. So the healthy cell is not going to be affected by the infection. Now, if you go into a cancer cell, and the cancer cell has no antiviral mechanism by interferon pathways, then the cell is going to die. Okay. Now, the problem we have with that type of virus is that it is easily recognizable by the immune system. So the immune system, the immune system is just going to kill it, not knowing that it's a good thing that is killing the cancer cells. The immune system is just going to get rid of every cell that is infected, and then get rid of the virus. So if any cell was not infected that is a cancer cell, it's just going to grow again.
So what about suppressing the immune system on one?
When you suppress your immune system and you put a virus into your system, that is not a good idea. So you have to have a balance between how much you want to suppress, usually you don't want to suppress it, and how much the virus is going to work. There are ways around it for example, with site-directed mutagenesis of specific proteins of the virus, there are ways around to fool the immune system into helping the virus to do its job. But that's,
like the lawyers are like, basically signals on it so that it looks like it recognizes it, like tricking it to believe that it's a normal cell or a cell within your body.
it's basically not activating the immune system to the extent that the wild type would. So, but
Do you usually think it's more effective? Because I've been doing research on plant-derived compounds that they've been injecting the tumor cells and eradicating tumor cells, Allison from virus. So, do you know which one would be better, viral treatment or a plant-derived compound?
That's a hard question because it depends on the cancer cell. Some cells are going to respond better to a chemical, some other cells are going to respond better to a virus, some cells are not going to respond to anything. So there's no one fix, one size fits all. There is no such a thing. As I mentioned, there's not going to be one cure for cancer. You might have to take one cancer at a time and see which one works. Now, the idea of the virus bursting out the cancer cells, that's a good idea because about 80 to 90% of all malignant cancers have defect in the interferon pathway. That's a huge percentage. And those are cancers that are very difficult to treat because they are malignant and they're traveling in the system. They go to other organs, they go to the brain. So if you cannot catch that cell, if a virus can go in there and catch that cell, it's a big plus. Okay. Okay. So we talked about the repair mechanisms, right? We talked about nucleotide excision repair, base excision repair, and mismatch repair. Then we had the lecture on how we avoid developing cancers by use of these DNA repair mechanisms. Hope it's still fresh in the mind. Okay, now, when you have a cell, a normal cell, at one point, not everything, nothing will live forever. So every cell has its time to, divide and grow and die. So there are two ways in which a cell can die. One way is via necrosis, and this happens when you damage the cell. Like, for example, you're riding the bike and you fall off and you just, burst a few of your muscle cells and you get all this necrosis, looks all black, and got with some blood going around and some inflammation, and it hurts like hell. And a few days, okay, the immune system or your phagocytes, whatever, take care of the inflammation, and then you reconstitute your skin cells, you're fine. So that's the necrosis type of cell death. Now, the other type of cell death is the programmed cell death or apoptosis. And the cell can undergo apoptosis by one of two different ways. Apoptosis, what happens is that the cell will just implode on itself. It will destroy itself from within and will not cause inflammation. This is a very important distinction between necrosis and apoptosis. So if you want to compare some analogies, if you have necrosis, it's like you have an explosion. And then if you have apoptosis, it's like you have an implosion. Think a building, necrosis, you explode the building, and then apoptosis, you just implode the building. It does not involve the areas around the building. So how do you cause apoptosis? You can have different types of signals. You can have signals that come from outside of the cell, or you can have signals that come from inside the cell. Now, from outside the cells, it usually involves signals from the immune system cells. Those are signals that come from natural killer cells, the cytotoxic T cells. They have these specific receptors, the Fas and Fas-ligand receptors, that will tell a cell that it contacts, it will tell the cell to commit suicide. That is the outside signal. Now, if you have signal from inside the cell, what is that saying? That's saying that if the cell notices some kind of DNA damage, like when you have mutations, UV light damage, and the cell cannot repair it, so the cell is going to trigger the signals from within the cell, and is going to go through this mitochondrial pathway, you're going to have cytochrome C leakage, and the cell is going to still die via apoptosis, but it's going to be from the inside. It's not going to be from outside the stimulus. So then, you have events that are proteins that are important for anti-apoptotic effects, and proteins are important for apoptosis. And you can imagine if proteins that are very important for anti-apoptotic effects, that means it will cause this cell to be more resistant to apoptosis. It's not going to cause the cell to be vulnerable and die. Those genes have to be, tightly regulated. You don't want the cell to die very early, and you don't want the cell to live forever. Now, what else is used for suppressing cancer or triggering cancer to develop? As I mentioned, you have cell cycle control, you have DNA repair, you have apoptosis, and of course you cannot forget about those telomeres, the telomerase activity that helps the cell to live forever. Okay, we know all about telomeres, and as I mentioned also, where else is a telomere active? When you are in utero. Gametes, right? Any other cells? Gametes and cancer cells, yeah. I don't know of any other cell that has telomerase activity. Okay. And then it would be also a good idea if you could target the telomerases and get them to stop their function. If you stop the telomerase in the cancer cells, and the cancer cell is dividing very quickly, so it needs a very active telomerase in order to keep on dividing. If you stop their activity, the cancer cells are eventually going to die. Yeah, actually, I don't think anything is bad about this because all the other cells don't have active telomerase anyway. Yeah, you always have some type of side effects. The only problem is that there is no such a drug yet. So I think people are thinking about for telomerase. No, there are no drugs for telomerase. Not that I know of yet. But soon, I guess, they will have something. I'm sure somebody's thinking about it. I'm sorry? Yes, they would. Pretty much. But you have always ways around it. Because if you freeze, woman freeze the eggs or freeze some sperm, the technology these days, is amazing. So you can always, if you have to make a choice between surviving without cancer and thinking about having a child later, maybe the choice is clear. Now, this is the translocation that I had mentioned before. Okay, that some chromosomal instability is very general, it's very common in cancer cells. And there is this one translocation between chromosome 22 and 9 that happens in all these cells that cause chronic myelogenous leukemia. Okay, and also remember that when you have cancer cells, if you keep in your head the idea of the HeLa cells, when you're looking at the karyotype, you've never found those 46 chromosomes. You found hundreds or found few, or 3, 4, and they're very aneuploidal. So the chromosomes are totally messed up. So let me show you here a picture of first here the translocation, just a little picture of what this translocation looks like between the two chromosomes. Remember the bigger one has the smaller number, chromosome 9, and then 22 small. You have a shift here, translocation. This is, I don't know why it was discovered in Philadelphia for some reason. Yeah, but you see here, this is a reciprocal translocation for this Philadelphia. It's not only one way. I was wrong with that one. It's reciprocal. I was making confusion with some other gene here. The cause is also myelogenous leukemia, and another form of very malignant T-cell leukemia. But anyway, I'm not getting into that one. So this one here, yeah, this Philadelphia business, it's reciprocal translocation. So let me show you here how the chromosome pattern of a cancer cell would look like. You have trisomy in one part, another 3 trisomies here, 4. You have something here that who knows what in the world that is, but definitely does not look like a pair of nicely looking chromosomes. So this is what a cancer cell, and is this a man or a woman? Yes? How many people think it's a man? Oh, nobody? Nobody? Woman? Man? No Y chromosome there. Right? 2 X's, no Y. That's a woman. Oh, this is a cool part. This is a very neat part about what the cancer cells do in order to become malignant. So when the tumor is growing, when it's small enough, it will get the nutrients from the environment. But then when it starts to get bigger, it will need its own blood supply. So how is it going to do that? It does it by 2 ways. It will activate genes that are important for angiogenesis. Angiogenesis is the formation of new blood vessels. So it will start to activate genes like EGF, DEGF is a growth factor, epithelial growth factor, and those cytokines are going to stimulate the nearby blood vessels to start growing new vessels towards the tumor. And so you can imagine that if you have specific drugs that inhibit this process, then you might as well just kill the cancer cell because it's not going to get a good blood supply. Either you'll kill the cell because it's going to starve, or you're going to keep the cell, the tumor from growing, because it will come to a certain point that if it does not have a supply of nutrients, it's just going to stay there or die. So this is a good strategy for attacking specific types of tumors from developing. And one thing that is very neat about this is that the cancer cells, not only they secrete these angiogenesis factors, pro-angiogenesis factors, but they also secrete metalloproteinases, and those are going to degrade the matrix that is, the stuff in between the tumor and the blood vessel. Whatever's in between something, they call the matrix. So if you have a matrix here separating the tumor from the blood vessel, these metalloproteinases that the tumor will secrete are going to break down this barrier and is still going to attract new blood vessels to itself. So these cancer cells are really smart in the sense on how to get a blood supply. Oh, here, this is another very cool video that I think is important for you to see. I am not for a Vita Medical Center. The video is really neat. I have no financial, what do you call it?
And some of these mutations
interest.
may be due to carcinogens, toxins found in the air we breathe, the food we eat, the water we drink.
Okay, this is a very concentrated summary of the cancer lecture.
Some of these mutations may be due to carcinogens, toxins found in the air we breathe, the food we eat, the water we drink, not to mention products that we use. Also, the aging process may bring about genetic mutations, the metabolic aging process of each individual cell. Also, genetic mutations that occur simply due to the genes that are available within our bodies. Also, infections, such as viruses, can play a role and contributors to causing cancers. Once these mutations occur, even subtle mutations, cancers begin to grow and transform. And as these cells begin to grow, we develop tumors. Angiogenesis literally means blood creation. In order for a tumor to grow larger than the size of a pea, they need a blood supply to divert nutrients away from healthy cells. To accomplish this, tumors release chemicals such as interleukin-8, vascular endothelial growth factor, and transforming growth factor beta, which causes blood vessels to reroute themselves to the tumor, supplying it with all the tools it needs to grow and to spread to other areas of the body. Cancer cells enter the bloodstream using a process called metastasis. Metastasis is where cancer cells break away from the primary tumor and spread to other areas of the body. Metastasis accounts for 90% of cancer-related deaths. Once cancer cells enter the bloodstream, it can travel to anywhere in the body that blood flows. This includes the bones, lymph nodes, organs, and even the brain. Metastatic cancer cells can lie dormant for days, weeks, months, and even years, and then become reactivated. Micro-metastatic cells can go undetected by CT imaging, MRI, or PET scans. Currently, there are no chemotherapeutic agents specifically designed to combat micrometastasis. However, natural killer cells and natural killer T cells are the body's own innate defense against micrometastasis. Because of the effects of chemotherapy on the immune system, the proper function of natural killer cells and natural killer T cells is crucial. Questions to review. What are you doing about the carcinogens or chemicals or infections?
Okay, that part you don't need to know. That's propaganda for this Evita medical group. But this is a very nice, concentrated video about the tumor growth, about the angiogenesis factors, and about metastasis, and also about the idea that immune system cells are going to go after these cells that are trying to escape from the tumor site. Now, one last thing, before you go, talk about metalloproteinases, right? One thing here that is important to know: how is it that the immune cells are going to kill the cancer cells? Here, if you have a T cell, cytotoxic T cell, it has this Fas ligand at its surface. And then if you have cancer cells, usually they express this Fas receptor. So when you have a T cell that finds this receptor and connects the Fas ligand with the Fas receptor, it will activate this target cell. In the case of cancer cell, it will activate this cell to undergo apoptosis. Okay, so this is one way in which the T cell will kill the cancer cell. Now, unfortunately, the cancer cells figured out that if they just liberate Fas receptor into the environment, they can block this action from the natural killer cell, from the T cell, because if this will find Fas ligand out and it will bind it, then this ligand here doesn't have a function anymore. It's bound to something and it's not bound to anything else, like there is no cell over there, right? So the cancer cells will secrete these Fas receptors into the environment in order to trigger the cytotoxic T cells not to kill them. And worse yet, the cancer cells, a lot of times, will express Fas ligand themselves. So what does that do? When the cytotoxic T cell gets there, it also has some receptors on its surface, and when it finds the ligand on the cancer cells, it commits suicide, because the signal is going to go from the ligand to the one with the receptor. If the T cell is the one with the receptor, it will get the signal to commit suicide. So there are two ways in which the cancer cell can fool the immune system. One is by liberating this factor that is going to bind to the ligand, and the other one is by expressing the ligand itself and then causing the T cell to destroy itself. So you see that fighting the cancer is really a pain, right? So here, okay, I'm not going to get into population genetics.